Very good morning, dear students. The next topic in unit number 5, cyclic nucleotides. So we have been discussing nucleic acids and their component and the structural organizations, and the previous class we have discussed nucleotides, di and trinucleoside phosphates, and the next type of nucleotides are cyclic nucleotides. So these cyclic nucleotides are very important component in biochemistry because they perform several role in many metabolic pathways, and they mainly act as the signaling pathway as secondary messengers. So before we are going to discuss the details about cyclic nucleotides, so we have already mentioned a brief about cyclic nucleotides. So cyclic nucleotides are, it is a single phosphate nucleotide with a cyclic bond arrangement between the sugar and a phosphate group. Okay, the sugar and phosphate group, it has a single cyclic rearrangement. Okay, it's a single phosphate cyclic rearrangement. So as you all know, nucleotides, it's a combination or a main conjugate between nucleoside and a phosphate group. A nucleoside is an interaction between the nitrogenous base and a sugar. So when these nucleosides are interacted with a phosphate group, then it is known as nucleotides. And the cyclic nucleotides are, there is a cyclic bond formation between the sugar and the phosphate group. And the cyclic nucleotides are composed of three important functional groups. One is a sugar, next one is a nitrogenous base, the third one is a single phosphate group. And in cyclic nucleotides, the cyclic portion consists of the two bonds between the phosphate group and the 3' and 5' hydroxyl group of the sugar in a ribose or deoxyribose sometimes. So it has two bonds. So the bond formed between carbon number 3 and carbon number 5, okay, that has a hydroxyl group, and that hydroxyl group will be interacted with the phosphate. Then there is a formation of a cyclic bond. There will be a formation of two bonds, and it will be formed in the formed like a cyclic compound. So this is the structure. Okay, so this is the base, and this is the sugar, and this is the phosphate group. Okay, so this is a brief structure, and here you can see an example of a GMP. Okay, guanosine monophosphate. Okay, this is a cyclic GMP. Okay, so here you can see guanine, a nitrogenous base, comes under purine. Okay, and you know that this N9 and C1 bond phospho, that is a glycosidic linkage. Then we are having the C2, C3, C5. Okay, the C3, here there is a hydroxyl group, and C5 there is a hydroxyl group, and this hydroxyl group and this hydroxyl group will be interacted with the hydroxyl group that is shared with the phosphate group. Okay, here OH and here OH. Okay, then you will be having OH and OH. The removal of a molecule of water will take place. Then the oxygen will be shared like this. Okay, so this is the two bonds. Okay, one is 3', another one is 5'. Okay, so here you can see a cyclic ring formation. Okay, that is a phosphodiester bond. Two phosphodiester bond is produced. So this is the general structure of a cyclic nucleotide. And the cyclic nucleotides, there are a lot of studies have been conducted, the mechanism, proposal mechanism, and their interaction in the biological function in biochemistry. The role of cyclic nucleotides and their ability to perform physiological reactions, especially physiological signals in a cell, has studied in a glycogen metabolism by two scientists, Carl and Gerty Corey. Okay, for this particular study, they have awarded the Prestige Nobel Prize in 1947. Okay, so later, after really 40 years later, in 1985, the study has been continued, and the Cyclic GMP as a reported. Okay, and here the three scientists, okay, again, Evgeny Festenko and Stanislav Kolenskov and Arkady Lybarsky, okay, they have discovered the cyclic guanosine monophosphate, that is CGMP, which can initiate the photoresponse in the rod cell that is present in our eye. Okay, so in our vision cells, okay, that is rod cells and cone cells, you know, in that rod cells the photoresponse that is mainly involving. In 1988, the very important enzyme called adenyl cyclase that is identified, and which synthesize the cyclic AMP. Okay, which synthesize the cyclic AMP is regulated by the hormone and neurotransmitters, and also by phosphorylation, calcium, guanine nucleotide binding protein. Okay, so this enzyme is performing a very vital role for the production of cyclic AMP. So this is the brief history. And what is the biological significance of the cyclic AMP? The cyclic AMPs, their biological significance includes a broad range of protein ligand interactions. Okay, that they plays a very vital role in the protein ligand interaction. As I mentioned in the beginning, they act as the secondary messengers in hormones and iron signaling pathways in most of the eukaryotic cell. And they also play a very vital role in the allosteric effector compound. They act as the allosteric effector compound of DNA binding protein in the prokaryotic cell, especially bacteria. Okay, so that is very important in how microbiologically how these cyclic AMPs, or cyclic nucleotides, are playing a very important role. Then the cyclic AMPs and cyclic GMPs are currently the most well studied cyclic nucleotides. Okay, cyclic AMP and cyclic GMP. But cyclic CMP, that is a cytosine, it is in eukaryotic cells are also found to be involved in the cellular messaging and some other signaling pathway mechanism. And the discovery of this cyclic nucleotide has contributed the great understanding of the two important types of enzyme, kinases and phosphatases. Okay, their biological reaction, their biological enzyme action mechanism that is studied very well. In addition to that, how the protein synthesis is regulated in the body also has been studied. Okay, so that is the another important significance of cyclic nucleotides. Then these cyclic nucleotides, depending upon the nature of the nitrogenous base, there are different types. As I said, most well studied cyclic nucleotides are cyclic AMP or CAMP and cyclic GMP, okay, CGMP. But cyclic CMP and cyclic UMP, okay, are less studied, and there are so many studies are in progress. So based on this one, there are four types of cyclic nucleotides. One is cyclic AMP, that is fully known as 3' to 5', cyclic adenosine monophosphate. What is this prime? We will see. Okay, so cyclic GMP 3', 5', cyclic guanosine monophosphate. Then CCMP, cytidine 3,5 monophosphate. Then CUMP, uridine 2' and 3' cyclic phosphate. And as I said in the beginning, all these structures has three important component: nitrogenous base. Okay, so for example, in cyclic AMP, that is adenine, and cyclic GMP, it is a guanine. That is the main difference, the nature of the nitrogenous base. Then the sugar, most probably the sugar is 5 carbon sugar, that is ribose, than the deoxyribose. Then you will be having a phosphate, and this is the base, nitrogenous base. There is a sugar. The bond between these two are beta, a glycosidic linkage, and you can see this is interacted with the phosphate, and you can see the phosphodiester linkage. Okay, then we are going to study the structural aspects of cyclic nucleotide in detail. Okay, So here, as I said, the nitrogenous base are either purine or a pyrimidine. Okay, so purine, you know, that is there are double ring. One is a pyrimidine ring, another one is an imidazole ring. Okay, this double ringed purine is the nitrogenous base for cyclic AMP and cyclic GMP, but cytosine, thymine and uracil. Okay, each has only pyrimidine residues. The ring only one single nitrogen base ring, that is the pyrimidine. Okay, then this nitrogenous base is attached to the first carbon atom of the ribose. Okay, carbon number 1. Okay, the nitrous base is attached to the first carbon of the ribose sugar, and the phosphate group is attached to the fifth carbon of the ribose. Okay, that is another important thing. Then the phosphate group makes a second connection to the ribose ring at the third carbon atom in the cyclic nucleotides. Okay, so this is what I have told. Here you will be having the third one and the fifth one. Okay, so the phosphate is interacted with here you will be having the fourth one, and here you can see CH2OH, and here you will be having the fifth carbon, and here you will be having the third carbon. That two side main OH, you will be having the bond, then it will be having two bond formation. That is what I told. The phosphate group make a second connection to the ribose ring at the third 3' carbon in a cyclic nucleotide, and because of the phosphate group has two separate bond in the ribose sugar, it forms a cyclic ring. Okay, so this due to this bond, okay, and this bond, it forms a cyclic ring. Okay, so that is another important aspect. And the atom numbering is used to identify the carbon and nitrogen within the cyclic nucleotide. Okay, so in the pentose sugar, the carbon closest to the carbonyl group is labeled as carbon 1. Okay, the pentose sugar, carbon closest to the carbonyl, these things are labeled in, this is carbon 1. Okay, carbon 1, 2 and this is 3. Okay, so that is the thing. Then when the pentose is connected with the nitrogenous base, the carbon atom numbering is distinguished with the, so you can see the same numbering is there. When the pentose is connected to a nitrogenous base, the carbon atom numbering is distinguished with a prime. That is why what we have called as this primes, 3', 5'. Okay, so that prime notation which differentiate this carbon from the atom numbering of nitrogenous base. Okay, in order to differentiate the atom number from the nitrogenous base, so the carbon that is attached to the pentose and attached to the nitrogen base is denoted as a prime. Okay, so that is the numbering system. And although there are 3 different types of cyclic nucleotide, we are going to discuss the first 2. These are very well studied molecule, as I said. So cyclic AMP, that is 3', 5', cyclic adenosine monophosphate, and cyclic GMP, that is 3', 5', cyclic guanosine monophosphate. We are going to discuss in detail. First one, cyclic adenosine monophosphate or cyclic AMP. Okay, cyclic adenosine monophosphate. This is known as CAMP or cyclic AMP or 3' to 5' cyclic adenosine monophosphate, all those things. And this is identified by while studying the mechanism of action of hormone via secondary messenger. Okay, by a great scientist in that is known as Earl Sutherland. Okay, in 1971. He got the Preston Nobel Prize in Physiology or Medicine for this particular study. Okay, the mechanism of action of hormone by secondary messenger such as cyclic AMP. So it is a second messenger important in many biological process. Okay, this is a very important molecule that is important for many biological process. And cyclic AMP is a derivative of adenosine triphosphate, that is ATP. That is the primary energy source. Okay, you know that actually, and which is used to the intracellular transduction in many different organisms, which is conveying the cyclic AMP dependent pathway. Okay, so here you can see the structure of cyclic AMP. This is, you can see adenine. So this is the pyrimidine ring. This is the imidazole ring. This is the sugar. Okay, as I said, nitrogen NH2 is there in the case of this pyrimidine. sorry, in this case of adenine. Okay, so here N9, okay, N9C1, okay, C1N9 bond, beta glycosidic linkage, and here you can see the cyclic bond 1 and 2. Okay, so that is this is the phosphate group. So this is a 3-part nitrogenous base, the sugar, and the phosphate group. Why it is cyclic? Because here you can see a cyclic structure. Okay, that is the importance of cyclic adenosine monophosphate. And what are the functions of the cyclic adenosine monophosphate? So this, as I said, this is the most important thing of, this is the another important thing of CMP. Okay, it is a second messenger used for the intracellular signaling. Okay, intracellular signal transduction, which transfer the cells the effect of hormones like glucagon and adrenaline, which cannot pass through the plasma membrane, which transfer into the cell the effects of hormone like glucagon and adrenaline, which cannot pass through the plasma membrane, which involve in the activation of protein kinases. Okay, then the cyclic AMP binds to and regulates. Okay, binds to the channel and regulates the function of ion channel and cyclic nucleotide binding protein. Okay, it binds to the protein and which regulate the functions of so many ion channel that is present in the organism. So these are the function of cyclic AMPs. The next one, cyclic guanosine monophosphate or a cyclic GMP. Like cyclic AMP, this is very, very important. Okay, the cyclic guanosine monophosphate is very, very important. So CGMP is a cyclic nucleotide derived from guanosine triphosphate. Okay, that is known as GTP. It is derived from GTP. Okay, so cyclic AMP is derived from ATP, like that. So this one also acts as the secondary messengers. Okay, so the mechanism of action is the activation of intracellular protein kinase, okay, which responds to the binding of membrane impermeable peptide hormone to the external cell surface. Okay, so these are the important things. The mechanism of action is the activation of intracellular protein kinase, the response to the binding of membrane impermeable peptide hormone to the external cell surface. Okay, so this is the structure. This is the guanosine or guanine. Okay, this is guanine. You can see NH2 and O, and this is the sugar. Okay, this one, and here you can see the phosphate. Okay, so this is the cyclic ring structure. Okay, so this is the nucleotide, cyclic nucleotide. Okay, this is another important thing. Then what are the functions of cyclic GMP? Cyclic GMP is having lot of function. It is mainly involved in the regulation of some protein dependent kinase. Okay, so very important role it will be playing. That is I have already told you, for kinases this can perform a very important role. The common regulators of ion channel conductance, and it is act as the common regulator for ion channel conductance for main like glyco geonolysis. Okay, then cellular and other cellular apoptotic process. Okay, it's a common regulator for the ion channel conductance and glycogenolysis and cellular apoptosis, and which relaxes the smooth muscle tissues. Okay, it mainly involved in the relaxation of smooth muscles, and in blood vessel the relaxation of vascular smooth muscles, which leads to the vasodilation and increased blood flow. Okay, then it is the secondary messengers, secondary messenger in photo transduction in eye. Okay, the mediate the switching on the attraction of apical dendrites of pyramidal cells. Okay, apical dendrites of pyramidal cells. Okay, then the binding proteins and functions of cyclic GMP. The binding proteins and functions of cyclic GMPs. The cyclic nucleotide, the cyclic AMPs, the main binding proteins are protein kinase A, cyclic nucleotide gated ion channels, and EPAC. Okay, that's another important protein, and catabolite activator protein CAP. Okay, so these are the important protein. The main pathways are which are associated with this one: smooth muscle relaxation, photo or olfactory receptors, and glucagon production in pancreatic beta cells, and lack operon regulation in E. coli. Then similarly, cyclic GMP, there are two important proteins, cyclic GMP dependent protein kinase or PKG, then cyclic nucleotide gated ion channel. So this we have already seen the function. Then the biological pathway is smooth muscle relaxation and photo olfactory receptors. Then cyclic CMPs, the cyclic GMP kinase 1, the protein kinase A, the smooth muscle relaxation. Okay, so these are the important things. Then so with this we have come to an end of this particular topic, dear students. So once again we can glance through the main important topics studied in this particular class. We have discussed the cyclic nucleotides, okay, in general introduction, the general structure of cyclic nucleotide, the historical perspectives, the biological significance, the type of cyclic nucleotides, then the structure of cyclic nucleotides. The next one is cyclic adenosine monophosphate, and the function of cyclic AMPs, and cyclic guanosine monophosphate. Then we have discussed the function of cyclic guanosine monophosphate, then the binding proteins and functions of cyclic monophosphate. So these are the text and reference book. I told you already, Principles of Biochemistry by J.L. Jain is a good book, and along with the Leninger, Principles of Biochemistry. So these are the other reference book. So thank you, dear students, for watching this particular class. If you are having any clarification, you can ask me.